This is the fourth video of the series. I continue to explain the simple model of coronavirus infection. So, in order to understand the virus SARS2, is it engineered or not, you need to look at the natural adaptation pattern of other coronaviruses with a simple model. In the simplified example, let's say 1 million coronaviruses inhaled by pangolin or human or other organism, that doesn't matter. So these viruses hit the lung surfaces and they try to bind to sialic acid as their first interaction point. Among those viruses, 100,000 of them will be pulled into the cells by immune system receptors to be destroyed, to be digested. In this process, 1,000 RNA with damage gets out of these pockets of destruction, and these damaged RNA on their structural and functional sites of their RNA capable to transcript themselves. So amongst those transcripted viruses, they leave the first adapted host. One of them, if possible, by chance, with significant change, adapt to their new hosts. This is not only in coronaviruses, but many viruses. A signature event in this new host adaptation is to hide their sialic acid binding domain, the first contact binding domain, not to be detected by immune receptors in their ongoing adaptations and the future adaptations, future infections, and to survive or to adapt to their new hosts, to human hosts. But there's an exception. Severe acute respiratory syndrome coronavirus 2 does not have that qualification. It doesn't match to this model, simple model. So this virus is used. I would like to explain a bit the lung surface. So there are those structures here are receptors. They're outside the lung surface. This is the lung surface over here, and you can see the sialic acids are coated the surface. But in specific with SARS2, we have these ACE2 receptors. Those are anti-receptors, and also we have these furin that they will aid the infection. So in natural conditions, in previous coronaviruses, this is what happened. So those coronaviruses, they were detected from the bats, and they were detected by those immune receptors localized over the cell surface, and they were harshly processed because they did not have perfect binding capacity to their receptors, and they didn't have all except MERS, didn't have this furin cleavage capacity. So in SARS2, the pandemics we live, the actual problem is this: those defense system is offline. The virus is just coming here, perfectly sliding over here and getting into the ACE2 anti-receptors very easily. So at the next video, I'm gonna describe a bit more about this. Thanks for watching.